Okay, and here's the overview of this Jurassic Park activity. It's mostly videos and pretty self-explanatory, but let me just run through a couple of things. So this first video, if you watch it, they talk about how Jurassic Park came about and how they used DNA that they found in these fossilized mosquitoes to recreate dinosaurs. Okay, so hopefully you've seen the movie. If you haven't, you should go watch the movie. I know it's old, but it's a great movie. And some potential flaws, I'm sure you came up with a ton of them. But what I wanted to point out is that, first of all, a frog is not the best organism to fill in the holes, right? Because a frog is very distantly related. And if you look at the phylogeny, right, amphibians are right here. So even we are closer to dinosaurs than amphibians are. They could have used human DNA instead of amphibians. And so if we really want to use the most appropriate organism, it would be somewhere either birds or even crocodiles as a fill-in. Okay, and we're going to learn more about evolution and how to read these trees later, but just looking at this, it's pretty self-explanatory that amphibians are very, very far away. And so then we've got some opinion questions. So then Jack Horner starts to explain that you just can't get DNA from dinosaurs because they're just too old, right? And so if you go through this and you look at this Nature article, the half-life of DNA is pretty short, 521 years. And so after 6.8 million years, there's just nothing left. I'm sorry, after 1.5 million years, you really aren't going to get anything left. And so we don't have DNA from dinosaurs, but we do have DNA from the Neanderthal. And so this is kind of interesting. They actually asked for women who would be adventurous enough to birth a Neanderthal. That has not happened as far as I'm aware, but we do have the DNA full sequence, and so we could bring back a Neanderthal. But like I said earlier, just because we can doesn't mean we should. So anyway, and then we talk about this one where he talks about how it's closest to a bird. And so you should have chosen, a peahen would be a great organism to use. And then how did dinosaurs change so drastically? So if you read that article that I sent you, it talks about all the different things that may have happened, but the biggest hypothesis is this KT boundary that an asteroid hit. And we actually have evidence of this particular element that we don't see anywhere else on Earth at the KT boundary all over the Earth. So it suggests that it was a worldwide phenomena that actually spread this ash all over the place, and so it likely led to a very darkened sky, less photosynthesis, decrease in the food available for dinosaurs, and so these large, huge, massive energy-consuming organisms were just not going to make it. But they didn't die, right? The little ones survived and continued to evolve and became what we now see as modern-day birds. So kind of cool. And then he talks about how do we make a chicken into a dinosaur, which is so neat. And so one of the things he talks about is this atavism activation. So in our genetic code, we have leftovers from our evolution. So you saw from that phylogenetic tree that we're very much related to reptiles, and you're reading your inner fish, right? So we have sequences that may develop gills, or that may develop a tail, or that may develop a milk line, extra nipples all the way down our abdomen. And occasionally you'll get a human that's born with these funky things because there was a small mutation that reverted it back to functional. So it was probably a small mutation that made it non-functional. And so just a single mutation can bring it back. And so that's what they're trying to do with the chicken, is they're trying to go in and find those sequences that are no longer being expressed, but are still there enough that single mutations, small mutations, can bring them back and make them be expressed in the dinosaur. So things like teeth and claws and a tail to make them extra cool. And then we talk here about how does the body itself regulate what genes are on and what genes are off? Because obviously our cells are very specific, right? So when we're born, when we're growing as an embryo, all of our cells are what we call pluripotent, meaning that every piece of DNA is being expressed. The whole book is open. But as we go farther and farther along in development, signals happen between the cells that tell it, hey, you're going to be a heart cell, and you're going to be a skin cell, and you're going to be an intestinal cell. And so parts of the genome get shut down permanently, right? And they become determined cells. cells that can only be a certain cell type. And so some of those on-off switches we've discovered and been able to manipulate in some cases. So one of the on switches is what we call histoneacetylation. So these are the histones, it's protein balls that the DNA is wrapped around. And if you put acetyl groups onto them, if the cell puts acetyl groups onto them, it makes the DNA stay unwound and open. And when it's unwound, then the enzymes are reading it. Okay, so that's one of the ways that we turn genes on or leave them on. Another way that we coordinate gene expression is by transcription factors, which are also coded for by genes, right? So it's crazy. It's like this huge perpetual feedback loop, right? Where one gene turns another gene on, which turns another gene on, etc. But these transcription factors are present in certain cell types, which will make certain genes turn on and certain genes turn off, right? So like you can take, what's the name of it? It's not beta carotene, but there's another gene. I can't remember the name of it at the moment. But if you put it into the developing brain, for example, of a mouse, or no, not a mouse. That's not a good example. Something that doesn't have as developed of a brain. A mouse would actually work. So a mouse has much less folds in beta catenin. That's the name of it. A mouse has much less folds in its brain, which is a good sign of intelligence, whereas humans have lots of folds, lots of surface area where the neurons can grow, and so we are much more intelligent. And so you can actually take a mouse brain and bathe it in beta catenin, which is a transcription factor for making those folds, and you can make a folded brain, which I guess theoretically they would be more intelligent, but they in the experiments I've seen, they always sacrifice the mouse to look at the brain. So we really don't know. But anyway, transcription factors will turn genes on and off, which is really cool. And then ubiquitin is another thing. Ubiquitin is a way of getting rid of the finished protein product. So this is not at the gene level, it's at the protein processing level. And it's a 3 strikes you're out rule, right? So if a protein is done doing its job and it's just sort of loitering, I guess is the right word, then it gets tagged. And if it gets tagged 3 times, then a protease will come and chew it up. And so that's when we get rid of proteins. And then the last thing is methylation, which is turning genes off. And this is often the function or the mechanism that permanently turns off large parts of the genome in our different cell types. And that's where a methyl group is attached to the backbone of the DNA, and it does not allow it to unwind and be read by enzymes. And so often methylation is permanent, right? So for instance, nerve cells. We know that brain cells or the spinal cord cells don't regenerate because they've lost the ability to do mitosis, right? They've gone into G0, they've gone into retirement, and part of that is methylation of those genes required to go through mitosis. And so that's an active area of science, is can we demethylate these permanently methylated areas to bring the cell back out of retirement and grow new brain cells or nerve cells. All right, so that's just a little summary of what that is. And then this was just, wouldn't it be cool to have a chickenosaurus? Okay, so that's the overview for this activity.